in the jelly electrophoresis conditions. So another thing, let's talk about the RFLP, or restriction fragment length polymorphisms. It's a mouthful of name. Well, so restriction fragment means the fragment is generated by the action of restriction enzyme. Length means it's a length of nucleotides, and it's polymorphisms. Morphisms means the morphology of the DNA, and is poly means the morphology comes from different sources. So it's a different morphological lengths of the restriction fragments. That's the actual meaning of that. How can you achieve different lengths of restriction fragments in addition to add a simple restriction enzyme? For example, take two individuals. We have individual 1, we have individual 2. So in this individual 1 it has its DNA, individual 2 it has its own DNA too. So if we add a specific amount of restriction enzyme and the same type of restriction enzyme in both cases, for example we run echo R1, for example echo R1 in both cases to make the work possible. So we use echo R1, and for example we use BAM H1 in this case also. So we are using BAM H1 because in normal cases, in case of these eukaryotic cellular DNA breakdown, we have to use many different different restriction enzymes to cut them off, and also use HindD3. So HindD3 is another. So this 3, for example, we use these 3 types of restriction enzymes and use the exact same amounts. We take the exact same amount of the DNA. So this is their long DNA in both these cases, and after that, let me change the color. For these people I make the DNA straight away like that, and for this I make it another color with this green color, which is my favorite again. So we have two different individuals. We use the same restriction enzyme to cut them. So after using this enzyme to cut their DNA, what we find in this individual 2, we find the length of it is like that. We end up with 4 different segments. How can we know we end up with 4 different segments? Well, by running jelly electrophoresis. So we call them the restriction fragments now. So we have restriction fragments. We use the blotting technique to identify those genes. So we finally find what we called restriction fragment, or we finally find a blot. We will do blot later, but now let's think about the individual 1. In the individual 1, using the same sets of enzyme, we'll end up to form these divisions. Okay, so the same sets of restriction enzyme, but they make different number of copies of DNA. As we know, a restriction enzyme can cleave at the specific site of the DNA. As we know, in between two individuals of human, we have a very much similarity, above 99% similarity, and that suggests us it is 99.99% similarities between two individuals. But that means there is also 0.01% dissimilarities between them, and this experiment suggests us that among those individuals they have some dissimilarities too, dissimilarities in their genome construction, and small variations in their genome construction leads to the different sequence in their genome, and as they have different sequence in their genome, the same type of restriction enzymes will cut the different length of their genome. That's how we can find in this electrophoresis. So in the blot of our electrophoresis, if we run this blot in our electrophoresis, for example this is for individual 2 and this is for individual 1, if we run this electrophoresis here, this is minus, this is plus, so this is minus, this is plus. We run this electrophoresis. After running it, when we found in this case, we found this is a big part, a smaller one, another smaller and smaller. So we find 4 segments in different 4 length places, and in this case what we found almost this to almost similar range, another is this range. So that will be the dissimilarities between their DNA structures. We call them restriction fragment length polymorphisms, or RFLPs. So no matter how much we related to each other, no single 2 persons are similar genetically. They have some differences in their genomic content. This type of experiments is going to suggest us that. Okay, and we can determine with RFLP technology, restriction fragment length polymorphisms. So remember, so for example, we need to look at this, which for example we need to study or need to distinguish between two different individuals. How can we do it? We can do it using this restriction fragment length polymorphisms. We can take that DNA sample, cleave with different the same type of endonuclease, and after we can run it, and after the running we can find it. And not only in case of two individuals, but we can also find out a disease with this. How can we find disease with this? For example, in case of sickle cell anemia, there is a change between the one amino acid sequence, and as we know, the change in one amino acid sequence means there is something change in the base substitution takes place in the genomic level, and due to those base substitution, if we add different enzyme that will cut at the substitution region, we'll find different fragments, and from those different fragments we can determine whether a person is having allele for that disease or not. We can determine with that. So this is also important.